Welcome back to the GEP Pathways video walkthrough. This is a continuation of the Pathways video walkthrough after section 2.2. Now we'll be covering how we can determine the best collinear set of alignment to determine the potential location of Reb in Drosophila jacoba. As we can see, I have our prior BLAST results pulled up here on my screen, and we were able to determine that the best result was actually the very first. This was due to e-value, percent identity, along with understanding the differences on query cover as well. To begin by grabbing these collinear set of alignments, simply click on Drosophila jacoba strain Ti18E2 chromosome 3R. Once we've done so, you will notice that it goes ahead and provides 7 distinct matches in return. These are going to be 7 ranges that are aligning with our query and subject as the best potential matches based on this BLAST result. To have the best potential view of this and to really see how a pattern is forming, simply change the short by e value to the subject start position. Or in this case, now notice what this does is this provides us a particular set of all of these ranges in a format where we can see the query and subject all within a set boundary area. Now, this is going to be where one of the very first tables in our walkthrough is kind of coming into play. And to make sure that we all know how this table works, I'm actually going to go ahead and fill out part of it here as well. Notice that I actually already have it opened here, and we have our table 1 summary of our T-BLAST-N search results for the best scaffold match. Well, what do we know? We know as of right now we only have 7 distinct hits. So what I'm going to do is I'm actually going to insert more rows on this table so we can go ahead and have exactly what we expect. Now, once I have this finished, notice that I'm just going to go ahead and fill out each individual range that is distinctively directly found within BLAST. So we have 7 distinct ranges. So I'm going to go ahead and put in those distinct ranges. Make the easiest portions here for grabbing in that case. As you can see, we have range 1, range 2, all the way over to range 7 in this case as well. Now, what this will be able to provide is I'm going to go ahead and move this simply over here to the right. And I'm going to go ahead and shrink this over here to the left. When we shrink this over here to the left, we will actually be able to see pretty much everything exactly how we need to see it. But it's just going to be a little bit of additional navigation, but that's okay. So our query start is actually going to be this region here where it says query start 6. Notice that I could put 6 here, and our query end, we're just going to want to drag all the way over, and that 44 is where everything ends. Just like we had underneath for our subject, we have our starting region here, and I'm just going to paste that. And we have our end as well. So we have our subject start, we have our subject end, and most importantly, we also want to get the E value, identities, and subject frame. Well, maybe going ahead and doing it about this way is one way we could, but what about another way? We could simply grab the data just like this, right? I can go back to our NCBI BLAST just to show you many different ways we can do this. So we have an E value, right? We have our particular matches and our hits, right? Our expected value is 0.02. That's really not that great. But that is giving us some information, isn't it? I'm also going to go ahead and pull our percent identity, which is a 46%. And then last but not least, we want the subject frame. The subject frame in this case is going to be indicated on here as -3. Notice that it'll tell you frame right here as well. And surprise, we're just going to go ahead and continue this process all the way down. So why don't we go ahead and focus on range 2 together, and then we'll go ahead and fast forward once this table is completed. So for range 2, it's going to be the exact same thing. We have our query start, which is showing as 18. And I'm just going to clean that up a little bit. We have our end. Now, it might be easy to look at that 58 and say, well, that's clearly where it ends. Not quite. We want to go all the way down this range. So we have our query, and it ends at 108. So once we're here, it'll be our query ending at 108. Then going to go ahead and grab our subject, and we're going to do the exact same thing. We have our subject, we have our subject end right here. Remember, don't fall for the little trick just because it's end. We don't want to do that. Perfect. So I'm just going to go ahead and fill that out. Then we have our E value. So we're starting to see something a little better. Our identity is 28% on +3. So just like that. So 28%, and then this time we're on +3. So I'm going to take a moment to fill out the remainder of this table. And once I filled out the remainder of the table, we will be able to determine our potential best collinear set alignment. With that being said, I will be right back. Excellent. After going ahead and completing the entirety of the table, let's go ahead and take a look at what our final results are in a little bit more of a concise fashion. Notice that I have all of my ranges 1 through 7 located here, and I also have all of the query starts, query ends, subject start, subject end, e-value, identities, and subject frame. We've gathered all of this from our singular BLAST result. Now, what we're attempting to do is essentially answer the question, what coordinate in the target species gives the best collinear set of alignments to the protein of your target gene? Notice that that's exactly what it's asking for. It's also looking for you to summarize your findings from part 2 by recording the following information from your target gene in your species, which would include the scaffold accession number, and the approximate coordinates as well. So let's go ahead and take a look at what we have here. By going ahead and examining these individual regions, we can immediately take a look that there's going to be a little bit of some interesting things going on here. Let's take a look at range 1 and 2. If we look at ranges 1 and 2, range 1 is providing a negative frame with a really bad e-value. So if it were me, I would definitely go ahead and take a look at this and say, well, something is really interesting is going on here. Not to mention if we take a look at range 2, it's only covering 18 to 108. It's not covering 1 through 182, so a chunk of this seems to be missing, along with a fairly poor e-value and identity as well. So why don't we just go ahead and take this, and just go ahead and change the color there. And I'm just going to black this out just as an example here. So if I go ahead and take these particular areas, right, and I'm just saying like, well, what else could we go ahead and do here? Or let's just go ahead and say those aren't exactly what we're looking for. Let's take a look at our other ranges. We have 3 through 7. Notice that 3 is providing it start at 1 and ends at 20, but the e-value is 2e to the negative 78, and we also have an identity percent of 90% on a positive frame. If we look at 4, it's also providing us the exact same thing, 16 to 45. It's almost like it's taking what we had on query end of 1 and moved up with the next. So it's 16 to 45. Same e-value, 2e to the 78 with an 83% identity on the same positive strand. So that's all good news. If we go ahead and take a look at the pattern that's forming here. 16 to 45, 40 to 109, 111 to 153, 153 to 182. So what this is ultimately giving us is this is the best collinear set of alignments here. We can see that what we have is our query start from 1 to 182, which was the size of our BLAST query. We can verify that by navigating over to BLAST, scrolling up, and look at that. Our query length 182 matches up. There is a match there. Now, what we can see here is since we've organized everything in terms of our subject, we can see that our potential collinear set of alignments is going to be 191, 50809, all the way over to 191, 51, 699. And that is more than likely what is going to be our potential collinear set. So just to make sure that I really drive that home here, I'm just going to go ahead and I'm not going to turn that green. I'm going to go ahead and make that small change first and foremost. What I can do is I can go ahead and change the fill. So what we're looking at is our start and our potential end. Now, remember, this is not going to be our exact coordinates. This is just an estimate because the basic local alignment search tool is providing us that estimate based on other information it's able to locate. So what we could really see here is we could see that it is starting at 1 and ending at 182. Therefore, this range, 3 through 7, is actually providing us our best collinear set of alignments. So that is what we really want to go ahead and take away from this. So now that we were able to go ahead and do all of this in part 2, we were able to grab that translated protein. We were able to toss it into BLAST in order to determine a potential location. We were also able to go ahead and find the collinear set of alignments as to where our possible REB is in Drosophila jacoba. We will be able to continue with the video in order to determine what our next steps will be in part 3. Now, if we look at part 3 particularly, what is this going to go ahead and entail? This is going to entail examining the genomic neighborhood in our punitive ortholog.